Today's lecture will be on chapter 23, the blood vessels. So starting out the presentation, the first thing that we should look at is the structures of the arteries and the veins. There's many similarities to these. There's just some slight differences which we'll point out. So the first area that we're going to notice is going to be the tunica intima. The tunica intima is composed of simple squamous epithelial tissue. It actually makes up the lumen, or the hole in which blood can travel. In the venous system, what we see is the tunica intima has extensions on both sides which form one-way valves. Underneath that, the next layer, what we have located in both is going to be the subendothelial layer. So the subendothelial layer, its importance is it's made up of areolar connective tissue. And then we see the tunica media. This is where we start to see the beginning differences with this. So in the arterial system, what we're going to notice is that the arterial system has an internal elastic lamina. This internal elastic lamina is important for the rigidity. We don't see the internal elastic lamina in the venous system. The tunica media, when we're looking at the tunica media, it's innervated by the sympathetic nervous system. It causes the smooth muscle to vasoconstrict. And we can see the difference when we look at the tunica media in the venous system, how small it is in comparison to the arterial system. Again, we see an external elastic lamina. It's only found within the arterial system. Again, this allows for vasoconstriction and vasodilation. So we see that we have an internal elastic lamina and an external elastic lamina. The tunica externa, which is located in both, you can see the difference, however, is going to be the tunica externa. Its main job is to have the structure called the vasa vasorum here. So you can see the vasa vasorum is a network of blood vessels on the outer layer of the arteries and the veins. And you can see that the vasa vasorum, or where the blood vessels are lying, are much thicker in the veins versus the arterial system. And the last part, when we look at the capillary beds, we're going to notice that it's primarily composed of the tunica intima, which forms the lumen. And it also has a basement membrane, which we'll talk about as far as how things can dilute or pass through the different types of capillaries. So just some general characteristics when we're looking at the venous versus the veins versus the arteries. They both have the same. So as far as size is concerned, you'll have an elastic artery, which is the largest. You'll have a large vein. And then the further away you get from the heart, so as we're traveling down away from the heart, you can see that the size of both the veins and the arteries are getting smaller in diameter. When we are looking at the veins, it's a blood reservoir. It usually has about 60% of the total volume of blood when someone's at rest. We have all the working components. You have the tunica intima, which forms the valve, the media, which is going to be a lot smaller in the veins versus the arteries, and the externa, which has a more extensive blood reservoir, blood vessel system in the vasa vasorum versus the arterial system. On the artery side, we have the conducting portion, which means that we're looking at the elastic arteries. It's the largest of all three different sizes. So closer to the heart, we're going to find that the total diameter of the tunica intima is a lot larger. We find this areas such as the aorta, the pulmonary trunk, or the brachiocephalic. These are all considered to be elastic arteries. The next one we'll look at is the muscular artery. The most important aspect of the muscular artery is going to be the tunica media. You can see that proportionally this muscular artery has a proportionally larger tunica media than the other three. So the muscular layer is greater than the elastic artery. And the last one is the arterial. So what we see is the arterial is a lot smaller. This allows for the red blood cells to line up in a uniform fashion or a single file as it enters into the capillary. The arterial, what we see closer when we're looking at the arterial, it has the ability for arterial vasoconstriction, so it can reduce blood flow going into the capillary bed, or it can do arterial vasodilation, which increases the blood flow to the capillary bed. And then again, we have the capillary where we're going to see a true capillary where we're going to see exchanges of nutrients and minerals across the basement membrane. So a couple of interesting notes that we're looking at here when we're looking at the capillary bed. It's mostly composed, the endothelium is mostly composed of the tunica intima on both sides. So when we're looking at the tunica intima, we're looking at just the lumen. And when we're also looking at the tunica intima, we're going to have a couple of different structures that are important. The first one that we're going to look at is for vasomotion, which are these precapillary sphincters. So these are smooth muscles. So you can also see these smooth muscles. This would be coming into the capillary. So this would be the arterial coming in where we can vasodilate or vasoconstrict. So if we're vasoconstricting, then we're reducing the amount of blood flow that's going to come in. And this could be the end effect or an organ or a muscle or something like that. Or we could vasodilate, which would increase the blood flow going into the capillary. When we're looking at the true capillaries here, when you're talking about the true capillaries, you're looking at all the thoroughfares here. These thoroughfares, their job is to increase the surface area. It helps slow down the blood flow, allowing the nutrients, gases, and waste exchange in the effector or the organ. So the true capillary is just a way to increase the total surface area. The other parts that we're going to talk about are going to be the precapillary sphincters. So if necessary, these precapillary sphincters can all contract, and if they do contract, then we could increase blood flow coming back into the venous system, which would then return to the heart. So for instance, in low blood pressure or exercise, we can see these precapillary sphincters contracting to non-essential organs like the digestive system during exercise, and it'll cut off the true capillary system and allow for the blood flow to then return back to the heart to increase blood pressure. This is just another illustration when we see a contracted precapillary sphincter. For instance, this could be a capillary bed inside of the digestive system during exercise. It's not important to have the blood flow going to your digestive system. So on the arterial side, we would see vasodilation, and then we would see the precapillary sphincters contracting, which would cut off the blood flow going to the true capillaries on both sides of the digestive organ, which then would allow the blood to transmit quickly, still releasing oxygen and nutrients, but not as much. And then we see an increase of venous return for increase of blood pressure. When we're looking at the different types of capillaries, their main differences are going to be with inside or different components of their lumen or the tunica intima. So the continuous, this is the most common type. You can see here that we have the nucleus of the endothelial cell located along the edge here. Its endothelial layers are connected by extremely tight gap junctions. So here, these connected by tight junctions. These tight junctions help hold or maintain the rigidity of the lumen. We find these in muscles, skins, your thymus, your lungs, and portions of the central nervous system. And then when we get into the fenestrated capillaries, what we see is the fenestrations. You can see just these small little pinholes located here, here, or throughout the whole tunica intima or the lumen. This allows for fluid transportation between the blood and interstitial fluids. We see this especially in small intestines, allowing small micronutrients to pass through. And also endocrine glands when we're talking about hormone exchanges. And finally, the last one is going to be the sinusoid. And you can see that the sinusoid has not only large openings throughout the tunica intima or the lumen, but it also has a discontinuous basement membrane. So all of these, the basement membrane or outer covering is intact, and on the sinusoid what we see is the basement membrane is discontinuous. It's discontinuous for larger things such as like when we're talking about bone marrow, red blood cells can leave the bone marrow into general circulation. We see this with hormones, especially in the anterior pituitary, the parathyroid gland. We see this with the spleen, so fragmented red blood cells can make their way from general circulation in the spleen and get recycled. And also we see discontinuous membranes or sinusoids located in the liver. And finally, venous return. So when we were looking at the arterial system, it had a larger tunica media, which allowed for vasoconstriction or being under the influence of sympathetic nervous system allow for vasoconstriction or vasodilation. In the venous system, we have two different types of methods in which can influence venous return. One would be the skeletal muscle pump, and the other one would be the respiratory pump. So any contracted skeletal muscle is going to cause or form a squeezing action onto the vein, which then will propel the blood up into a chamber. And as it pushes up, then you can see here that we see the veins closing behind. This series of opening and closing will occur with all muscular contraction. Even the smallest muscular contraction will cause the blood flow to help return into the heart for increasing of blood pressure or increasing blood volume. The other way that we see are in two areas. One would be the thoracic cavity with the respiratory pump. So as you inhale, you're increasing blood flow into your thoracic veins. When you exhale, we see you're increasing blood flow into the heart and the abdominal veins. When you inhale, we see a decrease into intrathoracic pressure. And then when you exhale, we see an increase intrathoracic pressure. So that's going to be just in the thoracic cavity of how we're moving blood from the lower limbs up into the thoracic cavity. The abdominal pelvic cavity, what we see mainly is in inhalation, we see a contraction, which the contraction, when we see the contraction, we see that the diaphragm gets pulled down into a flat, versus relaxation when the diaphragm is is kind of concave shape. When we see the diaphragm contracting during inhalation, we usually see compression on the walls of the veins coming up, and we also see an increase of intra-abdominal pressure. During exhalation, we would see that compression released, and we would see a decrease in abdominal pressure. And finally, just a general overview of the systemic arteries. Mainly, we're going to focus on the ones coming up off of the top of the aorta. So on your left side, you have the left subclavian coming off and the left common carotid. And on the right side, you have the brachiocephalic trunk. So the brachiocephalic trunk is going to split into, or bifurcate or split. It's going to split into the right subclavian artery, and it's also going to split into the right common carotid artery. That's just a little bit of a difference between the right versus the left side. When we talked about the brachial plexus, it's important to note this is the axillary artery. So when you're looking at the brachial plexus, superior, inferior, middle trunks of the brachial plexus are all named in relationship to this axillary artery. As we go down, we have a descending aorta. Once you get into the descending aorta, you're going to have the common iliac, and then eventually the common iliac will then feed the lower half of the body. On the right-hand side with venous return, one of the most important ones is going to be the great saphenous vein here. This great saphenous vein is actually used for anyone needing open heart surgery. So they'll actually resect portions of your great saphenous vein and come up here, attach it to your aorta, and wherever the blockages occur in your coronary arteries. So that's just a little fun fact about the great saphenous vein. The biggest one coming out would be the femoral vein, which then turns into the external iliac, which then eventually connects up into the inferior vena cava. Again, as the blood returns, on the right-hand side, we have a right subclavian, and then it bifurcates into the right common carotid vein, and then we see it go into the right or superior vena cava through the right brachiocephalic. On the left-hand side, it's the same. You have the left subclavian vein here coming in, and then it's going to be dropping into the superior vena cava. So this was just a general overview or lecture of chapter 23. Thank you. Have a good day.